So
I
please.
just one other comment for Ron, and you know, in addition to measuring peak viral loads, or using that for your estimates, why not do the area under the viral load curve that you would get by studying infections in either humans or in non-human primates, and get some idea of the total amount of virus being produced during an infection. And again, that's complicated because we don't know if it's all infectious, and many of the clinicians think after a week, 10 days that the viruses that you're measuring by PCR are probably not infectious, and there are a few studies about that. But at least you could do the area under the curve for some period of time to get something a little bit more physiologically based. And also then when you're going into calculating the
yes, so I think that
possibly go on. I've done and published some calculations like that for influenza, where we were interested in the amount of virus being produced and the possibility of mutation. And the idea was to calculate the actual number of virus particles produced over the course of the acute infection, with the understanding that every viral particle is carrying an RNA, and each RNA, there's some mutation rate in producing that RNA. So anyway, we based it on the area under the curve, and then also tried correcting for the fact that what you're measuring is only a subset of the virus produced, because it's also simultaneously being cleared, at least in the in vivo situation. So you got to do a little calculation as to total production, which is slightly different than the area under the curve.
Yeah, so I will see if maybe one of the co-authors wants to also mention on that, but I think what you're suggesting is the more rigorous way to do it. Mm-hmm. The fact that we don't have the kind of developed models that we know your group has been developing through that makes it harder for us to do that, because it's much easier to us to do what we used to, like in this Fermi problem spirit, to do the multiplication and divisions. But if you really want to do the area under the curve properly, you need to have good models for that, which we don't. So it's really a question of that, and maybe we should do better.
Yeah,
especially if you think that this
you need a collaborator. I can just send you some references and make some suggestions as to how you might improve it, or at least at least recognize that it's a limitation of what you're doing so far.
I'll just say that we are currently using a kind of a crude version of the models that you're using, and it's basically highly dependent on the clearance rate of the virus, right? If the clearance rate is very high, then the number would be much higher than when it's low. So I'm not completely sure how well constrained are the estimates for the clearance rate based on the models. How sensitive are they to other model parameters?
Yeah, there are some estimates that people have published, and we've published based on trying to fit clinical data. But we know when the clearance rate is sort of the fastest time scale, which is, we know that's true for a lot of other viruses, then it becomes very insensitive. Typically, for human viruses, for HIV and hepatitis C, we've got minimal estimates that their half-lives are something like 45 minutes in circulation. And that's a very good estimate for C. There's estimates in non-human primates where you can infuse labeled virus or single cycle virus and sample very frequently. And there we've gotten clearance rates of order 5 minutes, half-lives of 5 minutes or less. There have been experiments again in non-human primates where you infuse known virus at known rates and try to get a steady, wait till they attain a steady state. And then you know the clearance rate out of the circulation is equal to the rate that you're putting it in. And also that comes out on timescales of minutes again for a variety of viruses. And so from clinical data, it's impossible really to get those types of clearance rates. And you're right, if they're very, very fast, then the total amount of virus being produced will be sensitive to that. But,
Alan, correct me if I'm wrong, but there's also the issue of the clearance rate out of the cell, like in the body, extracellular, and there's the issue of the clearance rate intracellularly.
Right.
And I think the numbers that you mentioned were talking about the clearance from the body, meaning extracellularly.
Yes, it's extracellular.
And I think for many of the things we're interested in the
Yeah, and actually the numbers I was mentioning are the ones that are easy to get, and that's clearance rate out of the circulation, and whether or not the virus is actually being destroyed in tissue, or some of it can be, especially if you're studying acute infection, some of that can just be redistribution until you sort of fill up tissue binding sites. So it's a little unclear exactly what that clearance is body-wide and what those rates are. From the easiest thing to measure and what is measured is out of the circulation.
But is there anything measured about the intercellular clearance rate? How fast, produced virions are getting emitted into the extracellular?
Yeah, very few estimates of that. We were looking at making complete models of viral life cycle, like John does for hepatitis C. And our best estimates there was that there was extremely slow, if not no, clearance within the cell. And there are some molecular immunologists, somebody like Ralph Bartenschlager, at University of Heidelberg, is a real experimental expert, and he sort of concurred with us that they don't see much degradation intracellularly, at least of hepatitis C. It's also a secreted virus, so same as SARS coronavirus, but I don't know very many measurements of that. It's something that you could do in vitro. John could possibly do that for the viruses he's looked at.
We haven't worked with hepatitis C, but we've got our hands full now starting on corona.
Yeah, no, it'd be interesting to look at, viruses that are, assembled inside and go out through the secretory pathways.
Yeah.
Our feeling was that once you, assemble a virion, it's highly unlikely that the cell would degrade it rather than just export it.
Yeah.
I mean, the RNAs intracellular, or the DNA intracellular
I guess
could be degraded, but once you make a viral particle, I think the degradation is unlikely to be very, very rapid or frequent.
Yeah, I might not be using the right term, but actually I would be curious to know if you, John, or anybody else I'm not talking about the degradation of a virion inside the cell, but I'm talking about the rate at which they're being secreted. Like for how long is the residence time of a virion inside the host cell before it's being excreted?
Yeah.
I guess then, so you would like to, you're saying that in principle, you can break open the cell and you can find infectious virions before it's secreted. That is possible, at least I know for phage systems that we've worked with. Phage T7 is a double-stranded DNA virus that you can break open the cells before they would otherwise lyse, and then you can capture infectious particles. So they are assembling within the cell and infectious prior to cell lysis. In other viruses here, we've got the viruses that are being secreted from the cell surface. Then they're not really virus particles until they have left the cell, in which case they're taking along some cell membrane. So I guess one couldn't really define a virus particle in that sense until it has been separated from the cell.
Yeah. In the SARS coronavirus too, I don't know if you've seen the transmission electron micrographs of it. I mean, they seem to fill up intracellular vacuoles, and they pretty much look like intact virus particles that are then being secreted.
Okay.
That's my understanding of it.
Do you know if people have isolated infectious virus particles from those vesicles?
I don't know.
Yeah, I know they're replication centers, but I wasn't aware of people. My sense is that they're probably very fragile, and it might be hard to do that, but it'd be interesting.
Yes.
So, I mean, one of the things that you both alluded to, there are several people in the room who do multicellular modeling. So instead of doing population level within the tissue viral loads, they're trying to model individual cells and their effects. And you had some nice, both of you had some nice statistics on variation in burst size between cells. one thing you didn't talk about, although you alluded to it, was the lifetime of the cell after infection. Something else one would love to have is the kind of thing, for example, that Jim Fetter would calculate, but all of, or last week, David Oda, which is the rate of viral production as a function of time after infection for a single cell.
Mm-hmm.
And I wondered if you could talk a little bit about what level of single cell data is possible. It may be that you don't need these single cell models, that the aggregates are enough, but there are certainly people in the room who would like to know, if you're going to model individual cells in the lung tissue, what kind of viral model should we have inside of each one, and how do we calibrate it?
You want to take that, Ron, or I'm actually looking for, so we did some work on single cell kinetics. And this is where you essentially dilute down you infect cells at high multiplicity. So these are not single virus particle infecting single cells. This is many virus particle infecting small number of cells so that all the cells get infected. And then it's to put those single cells in their own little wells with their own supernatant and to sample them periodically. So it's a crazy experiment, but I had a very dedicated graduate student, Andrea Tim, who had very good hands, and she did this. So let me see if, I'm not sure if I can share.
You should be able to screen share.
Okay. Yeah, so this is the paper from 2012. And so she was actually sampling multiple times supernatant from individual cells and replacing the supernatant with warm medium, and then allowing it to sit around longer and then sampling again. So she would get these, yeah, I guess this is the experimental setup in A. You make sure you have high multiplicity, so every cell you get is essentially infected. You dilute down, then you look for individual cells and individual wells, and then you sample and you replenish supernatants, and you generate plaque counts as a function of time from individual cells, and essentially integrating then the counts. This is your area under the curve, Alan, except for it's a single cell.
Mm-hmm.
Then you can get a total virus per time. So when she did that for, I don't know, this number of cells, 12 cells here, then in A, those are the single cell counts as a function of time. Then so that's PFU per hour, you could say. And then you integrate those and you generate these single cell one-step growth curves, which are all over the map with respect to yield, because we've got some that, here's one down here that's maybe 20 or 30 yield. And then here's one that's up around 10,000. These are all cells from the same dish. What's interesting, I think, also here is that the time is relatively narrow when these single cells are producing. The window at which they are producing is between maybe 5 and 8 hours, or maybe 5 to 10 hours. So huge differences in yields from single cells, but relatively narrow window in which they're producing. I think we did some additional analysis here. So you can look at various correlates here of rise times and latent times on this. So the longer the latent time or that you're waiting around, the higher the rise rate also tends to be. So I'm trying to remember going back to so we have these single cell data. I guess I'm now trying to revisit what was the burning question that we wanted to answer.
Well, again, I was saying for us, we have models of viral replication inside individual cells. We have a lawn of epithelial cells in our simulations.
Yep.
So we'd like to be able to, the aggregated data on net viral load in the simulation is not very informative about the microscopic parameters and variation between cells.
Yes.
And so, and if you're thinking about trying to explore therapies, that micro variation may be very important, and so you'd want to get it right.
Yep.
It's like trying to measure a tumor by looking at total tumor mass.
Yep.
It really is not a sufficient. The total viral load isn't sufficiently informative, perhaps, to be able to really. I mean, you could certainly build useful models, and Alan builds wonderful models that way. But if you want to do models where you model individual cells, then we need to be able to try to understand the survival time of the cell after infection, the production, all of these questions that Alan was addressing.
Yeah.
There's something else that you alluded to that came up. I heard a very nice talk by a Polish group that was looking at spatial correlations. In they were using a different not a coronavirus. And you were looking at interferon marker, a vital interferon marker reporter with a viral reporter.
Yeah.
And they were looking at correlations between interferon production levels, both RIG-I and STAT-P.
Yeah.
And they were different. Those reporters were very different. And the spatial correlations between infected cells, later viral production, and those two interferon markers. And they had rather complicated spatial patterns that were not what you would simply expect from a classic plaque assay where you have interferon getting ahead of the plaque.
Yeah.
They were getting very complicated, almost for a physicist, antiferromagnetic patterns where you'd get alternating high-low patterns. And I wondered what you were seeing if you were doing, and Ron, I don't know if you're doing those kind of spatial analyses, but when you look microscopically at spatial correlations in those kind of two-factor visualizations, what do you get?
Actually, I don't think I've seen anything like high-low, as you mentioned, antiferromagnetic, but it does remind me of the idea of potentially having Turing patterns. So, you know, when you have a diffusible activator and inhibitor, and the inhibitor has a higher diffusivity, then you can be generating dots or stripes and generating these spatial patterns in essence. And I wonder if that might have been happening because we have diffusible virus particles and diffusible interferons. And the interferon, the signaling molecules generally are smaller and faster to diffuse. So maybe there's something like that happening.
What they were seeing was actually, I think they felt I'm sure these things are very specific to the virus and also certainly to the cell type you're using in vitro. But I think what they were seeing was typically and again, it was particular virus. The virus had different ways of shutting down the two sides of the interferon pathway. And so depending on the race condition between viral proteins, particular viral proteins, and the two interferon pathways, you could get four states, for example, where one of the interferon path, the paracrine signaling was happening, but not the internal, and vice versa. So you had RIG-I versus the STAT happening independently of each other. And it was also affecting the way the virus was being shut down. And so I guess my point was simply that I think understanding that the levels was fascinating. What you showed at the single cell was fascinating, but looking at the spatial correlations would also be really interesting. And for the kind of spatial modeling a lot of people here do, it could be very interesting to try to replicate that.
Yep. Just the variability, one aspect here is the, moving away from ODEs to stochastic models, I think is probably something that is an approach that many have done also. I think Jacob might have had a question. Is that?
Joy, I'm sorry, I didn't mean to monopolize the conversation. Go ahead, Jacob.
I had a question. I put in the link. Someone answered my question. I just noticed it, but I put in the link and I'm looking at what you're doing and I'm not in that field. I do micro simulation, but like population models, but I'm trying to figure out what you're doing different than what I see is going on. So John Rice sent me this paper 6 months ago, something like that, long time ago. And I was looking at it, and this is actually only, this is for you maybe because the people that are mostly from Israel, you might know them. What's the difference between what they're doing, which is actually also showing different strains of viruses, and what you are doing? Because you're also saying there's a number of viruses and you're also counting them. What's the difference? Is it technique, or are you doing the same, or what's going on there? Just have a glance.
So I referred to that. I referred to that in my answer in the chat. But it's a very good paper and it's a very good group. We're in touch with them very a lot. And I would say what they did. We're doing different things in the sense that they're doing the hard work of really taking the samples, doing the sequencing, building the phylogenetic trees. We're doing the easy part of just taking some numbers and doing the multiplications and divisions and checking if things fit together. So they're doing an important empirical case study, and we're taking the more general back of the envelope calculation, seeing how things fit together and what we could generalize from that. So that's the overall general.
You're doing the higher level modeling. This is what you're telling me.
Yeah, maybe you could say that way.
Okay, this is your base data, what they're doing, or you have your own data?
No, so I've basically shown you the data that we're having. So we just it's not the same data in the sense that we started from very fundamental numbers, but then what we did is verify that the results that we're getting are in line with the results that they're getting. For example, the results that they're getting are also the result of selection. So it's not only neutral. So there's fine points here about what are you taking into account, only the natural drift or also the selection pressure. Necessarily in the real world, there's also the selection pressure, but often you find that this is how much does that change the order of magnitude of what you're doing? Often it does not in terms of those things.
You said that you are connected to them. Do you think that what they're doing will be interesting to this working group?
I hope so. Yeah. So the last author there.
I tried to contact them multiple times without success. Really, to all the emails. I looked up at all the emails. If you can, then can you connect them to James Glazier here?
Sure.
Thank
I'll
you.
do that.
Thank you. Then I'll be able to ask them the questions.
Okay. I see, Jim Fetter's on the call, Veronica's on the call. Do you have anything, maybe you talked to our speakers already without the meeting, but it's a good opportunity given to put some of the questions into the public if you'd like. I'm not trying to exclude anybody, but I just see Jim and Veronica as people who definitely work in this area.
Say hello. Though the very interesting discussion, we've been grappling with many of these questions too, obviously, and especially about this issue of how many infectious virions per virion, and the estimates that we've seen from our collaborators in line with this 10,000 number. But it is a little bit, it's mystifying, and I've seen there's some nice modeling work that suggests that these non-infectious virus particles can have a significant effect on the kinetics of an infection. That was work that Kevin Janes put out recently, his group, and this is very interesting to consider. But yeah, they're all. I actually did have one question for Ron, which was that about this issue. We haven't talked about it yet. The idea that 1 in 100,000 to 10 million lung cells being infected simultaneously, that seems really low to me, and it's very low. So we've so far only modeled infections in cell culture. So their MOIs are very high, and certainly not. If we told our collaborator to do a 1 in 10 to the 5 MOI, they would laugh at us, right? So how does this? How do you view that outcome?
So I view it with fascination, and if you can prove if you can prove us wrong, please do it now before the paper is out. I'll be happy to learn about it now.
Can I ask for a clarification on that? when you say 1 in 10 to the 5th cells, is that in an infected patch or is that over the whole lung? In other words, if I look at a plaque in the lung and you tell me 1 in 10 to the 5th cells is infected, then I'm nervous. If you tell me that 1 in 10 to the 5th 1 centimeter patches is infected, then I'm not nervous at all because the lesions are localized.
So what we calculate is overall. So we don't have a spatial model. So in that sense, it's like overall in the lung. And I'm guessing there's a lot of heterogeneity. So you're probably right. It's like in patches. But whether it makes sense that out of all the patches, you'll only have like say 10 to the -5, that I don't have a good intuition about that. I would mention that this value does become somewhat lower or higher, depending on how you want to define it, when you take into account only the fraction of lung cells that are expressing the, what is it, like the ACE2 and the TMPSS or whatever it is. So we've been taking that into account, and then it goes from 10 to the 5 to 10 to the 3, or 10 to the -3, if you like. Somewhere between, instead of 10 to the -5 to 10 to the -7 to 10 to the -3 to 10 to the -5. So still you need all this heterogeneity and patches and everything, but the values are somewhat different.
I mean, in the spatial models we do patch to patch, because we're simulating patches of a few thousand cells, there's a huge amount of heterogeneity. At certain level, even for the same parameters, some patches you'll get a few cells infected and the infection doesn't spread. Other ones, every cell is infected. Other ones, some fraction of the cells are infected. Now we tend to work, because we don't want to have to do that many replicas, with parameters where most of the cells get infected most of the time. But if you move the parameters just a little bit, you could wind up with a situation where 1 in 1,000 patches had a large rate of infection. Wouldn't have to move the parameters very much to get there.
Hi, I also have one question. So there are more and more papers appeared that showing that this SARS-CoV-2 could also productively infect human gut and enterocytes. So did you see any estimation? Because it's look like the virus production from those cells might be kind of much less, almost like go under the radar of the immune system. And that's what might even make them more like persistent for a while. Do you know anything about it?
Yes, so I don't know if that answers your question, but if you look at what I've just shared here, so in the digestive system, even though the overall mass is large, kind of like the lungs, the values that we found reported for the number of RNA copies or infectious units is close to the detection limit. So even though it exists there and you can find them in, say, in PCRs, etc., if you do the multiplication, you're orders of magnitude away. So we think it's a small contribution in the absolute values. That
So
being said, I'll mention again, this is based on rhesus monkeys. So if something is very different in the digestive tract of people in terms of the concentrations, maybe these results would also be different.
Okay, so it may be that they contribute to persistence by generated very small amount of virus, basically. Yeah, okay. Thank you.
Okay, well, I think it's been a it's our most successful discussion session so far. So I think we have to have, maybe we need Alan, Ron, and John back to have maybe a panel discussion to promote these conversations. Are there any last questions before we thank our speakers again and let them get back to their work? No, well, thank you both, and thank you, Alan, for coming.
Oh, you're welcome.
Really appreciate it. Thank you, all of you, and we really do appreciate your taking the time to speak to us and for the great discussion. And I hope we'll have
great
a chance.
Questions. Appreciate all the questions.
Okay, well, with that, I will call the meeting to a close, and I look forward to seeing people next week